Hello, my name is Saad Usmani. I'm the Division Chief of Plasma Cell Disorders at the Levine Cancer Institute in Charlotte, North Carolina. And I'm going to be talking about key considerations when approaching high-risk multiple myeloma. I think the overarching theme here is, despite the fact that we have a lot of newer therapies in myeloma, most of the survival outcomes benefit we've observed in the last 15 years is in our standard and intermediate risk patients, whereas the outcomes for high risk patients are quite poor. When we say high risk, what does that mean? This includes patients who have specific karyotypic abnormalities like deletion 17p, translocation 1416, 1420, gains and amplifications of chromosome 1q21. And one can consider translocation 414 as a high-risk feature as well, although many patients with the use of proteasome inhibition appear to be more intermediate risk. Then primary plasma cell leukemia, as well as elevated serum LDH twice above the upper institutional normal limit, tends to correlate with extramedullary myeloma. So if patients have any of those biologic features, we would consider them high risk. And certainly revised ISS stage 3 would kind of feature part of those kind of groups of patients. We only have one chance with these patients. We have to pick the therapies that give us the best likelihood of a deep response that we can then sustain. So, typically we would start with a PI IMiD-based induction treatment for these patients. The current guidelines based on the available data with Griffin and Cassiopeia we would consider adding daratumumab into that induction mix. For transplant eligible patients, they go on to receive a stem cell transplant, and then they can have a PI IMiD-based maintenance treatment approach based on data from the Arkansas total therapy days, from the Emory experience, as well as from the SWOG 1211 clinical trial. Then a case can be made of using KRD in select groups of patients who are unable to tolerate bortezomib as a proteasome inhibitor in those groups of patients. For transplant ineligible patients, it's typically IVD dose attenuated or light induction, followed by IMiD and PI-based maintenance. And then for transplant ineligible patients who are unable to tolerate a proteasome inhibitor because of neuropathy or other reasons, DRD or dara-Lendex would be a reasonable option as a combination chemotherapy. And then there are going to be certain instances where patients have bulky extramedullary myeloma or high burden plasma cell leukemia, where we're going to be using PACE or DSAP-based treatment regimens. But I would encourage younger investigators who are listening to this to think about developing smaller clinical trials focused on high-risk patients. This is a very different biology, their outcomes are different, and we're hoping that we can approach treatments in a different way in the coming years.